So next we're going to hear from Don McLeod, who's a non-commercial beekeeper with a background in the chemical industry. He's a member of the AP New Zealand, and is an advocate for bee health. He's also tag teaming with Dr. Chris Pook, who's a graduated from Exeter University with a PhD in marine ecotoxicology, actually. But he's also now employed as a senior research technician at Auckland University of Technology School of Science, Applied Sciences, and is running their analytical laboratory. He's an independent researcher with projects covering a diverse range of topics, including ecotoxicology of neonicotinoid pesticides. Excuse me. Can I please ask you to move in and grab a seat away from the exit doors, please? That would be most appreciated. So if you'd like to come to the stage now, that'd be great. Thank you.
Good morning, everybody. I'm Don McLeod. This is Dr. Chris Pook. We only met a little over a year ago, and I can't actually recall exactly who told me to get hold of Chris or go along to the AUT and see him, but since then we've had a pretty close relationship. We've been seeing each other nearly every month and recently every week. So things are moving along quite quickly. I'm not going to do a lot of talking. I'm going to be at the start and the opening and the closing. And we're going to deliver, we know neonicotinoids generate a lot of news. Today we're going to give you some good news and some bad news, and that there's a lot more to the news to come in the future. They're widely used in New Zealand, widely used worldwide, probably the biggest single insecticide in the world. One of the biggest earners for Bayer is the insecticide imidacloprid. They're very safe to handle, particularly for humans and mammals. They have very low vertebrate toxicity. They are systemic in the plant, and that makes them very effective. And that's why the farmer likes them. And they're effective on the sucking and chewing insects in particular. But they have some drawbacks. They're very persistent in the environment. We still don't know how persistent they are in all environments, and we've still got a lot of work to do to work that out. They control insects during the growing period of the plant, which is extremely important when they're used as a seed treatment to look after the seedling plant to get it established. But they do remain in the water and the soil for a long time. And interestingly, this year in our work with maize growers got a lower return for their maize this year, so where they were multiple cropping in the same paddock, a lot of them did not treat their seed because they knew there was enough in the soil to help control the insects. I probably should have titled this one, Is New Zealand Different? I got involved with the maize because of Neil Moss, up wherever he is. Where's Neil? Neil pointed out this problem to me down there about 4 or 5 years ago. I think it was at Napier. There have been no reports in New Zealand of hive losses at maize sowing. We heard yesterday about the problems in Ontario, but we have no reports documented about that from beekeepers during the sowing period and the dust problem that is experienced in Ontario. And if you have got the reports, show them to me. But I listen to the beekeepers, and when they tell me something, I believe them till we can prove it. We've got no reports during the growing season of hive losses. And this is particularly during tasseling when there's a large amount of pollen available and exudates from the plant. We have no reports during that period. And part of that answer was shown yesterday by Landcare with their spatial showing the food sources in the Wairarapa at the time of October, November, December, the period when there was a huge variety of food available. But beekeepers have regularly reported hive losses in the weeks immediately after maize harvest. And I can say from Paul Badger's work, there's 200 plus hives were lost in the Gisborne area in the last month. And we have called this maize flats disease. It's not a disease. So I'm going to pass you over to Chris, and he's going to talk about it, and I'll come back at the end.
Hello everyone, my name's Chris Pook. I'm a chemical ecologist from Auckland University of Technology. This phenomenon of unexplained hive losses has been going on for long enough that the beekeeping community has given it a name. We hear tales, I hear tales through dawn, of Gisborne flats disease, maize flats disease. We've set out to try and work out whether the clue is in the name. These neonicotinoid maize seed coatings, as Don's told you about, they're a very valuable tool for horticulture. The way they work is that the coating gets drilled, the coated seed gets drilled, sown into the ground, and the active ingredient disperses into the soil. The active ingredient is quite polar, it's water soluble, so it disperses freely into the soil and gets taken back up by the newly germinated plant and gets transported throughout the plant's tissues by its water transport systems. So that's a systemic component to this. And there, the active ingredient protects the plant against these biting, chewing, boring insect pests of maize plants. So how does that affect honeybees? Honeybees aren't notorious biting, chewing, or boring insect pests of maize. Well, the answer is the pollen. Honeybees love maize pollen. Maize is a good source of pollen for them when the plants are flowering, like you can see in the image on the right. Once a maize plant reaches flowering, it's old enough and big enough that the amount of active ingredient has been greatly dispersed and degraded. And so the amount of imidacloprid, in this case, that you can actually measure in the pollen of a maize plant is about 2 micrograms per kilogram. That's 2 parts per billion. I'm going to keep referring back to parts per billion to try and keep this in context for you and instead of jumping between different measurements. So 2 parts per billion, that's quite low, right? We can work out whether that's a threat to bees. We know that the lethal dose of imidacloprid is about 4.5 nanograms. We know that bees consume about 4 milligrams of pollen a day. And if you do a simple sum, you can work out that in that 4 milligrams of pollen, if they're consuming exclusively maize pollen, they'll consume about 8 picograms of imidacloprid. That's about 1/500th of the lethal dose. That's very implausible that pollen from imidacloprid-treated maize plants is killing bees. This is a dissipation curve of the neonicotinoid clothianidin in soil. And as I say, the active ingredient, once it's released into the environment, once it's planted, it starts to disperse and degrade. And you can measure the concentrations over time, and they follow this nice little curve, this exponential curve. So at the beginning, there are very high concentrations. You can see closer to the y-axis. But at the time of planting, at the time of pollination, at the time of flowering, which is well over 100 days into the life of the plant, the concentrations have fallen quite steeply. So maybe there's some mechanism occurring earlier in the season, which is when there are much higher concentrations of neonicotinoids present in the tissues of the plant and in the environment and in the soil environment. There's some other mechanism transferring those residues to the honeybee hives and causing toxicity. Well, this occurred to an Italian researcher called Girolami back in 2003, and he discovered that the guttation droplets, these liquid drops that are exuded from the leaves of young emerging maize seedlings, contain fantastic concentrations of the neonicotinoids. I was employed as a postdoctoral researcher back in 2009 to duplicate Girolami's work and expand it to more neonicotinoids than just the imidacloprid that he studied. So I ended up planting over 1,000 maize seedlings in the greenhouses at Exeter with treated and untreated seed of different neonicotinoids, and diligently collecting these little droplets of liquid that were exuded from the plants every morning. These samples got put into one of our fantastic analytical instruments, and we could measure, as Girolami reported, we found exactly the same thing. These are really, really crazy high concentrations of neonicotinoids, up into the hundreds of parts per million. Now, I spoke earlier about the concentration in pollen being 2 parts per billion. This is 10,000 times higher than that. These are concentrations which will kill a bee dead in minutes. Is this a problem? Probably not. The neonicotinoid seed coatings, the guttation fluid, does not seem to be systematically exploited, collected, by bees. There are anecdotal reports, and the picture on the right is a bit of a red herring. There are anecdotal reports of bees foraging and collecting guttation fluid, but it doesn't seem to be a systematic problem in New Zealand, because otherwise we'd be seeing massive reports of bee deaths around the time of seed sowing and the emergence of the sapling of the young maize plants. If we look at the calendar of the seasonal cycle, the problem which we are studying occurs in late autumn and winter, May, June, July time. The sowing time and the emergence time for maize in the north of the North Island is September and October. So if you plot the concentrations that you would expect to find in the environment of the neonicotinoids, they're at a maximum in September and October, and they quickly fall off as this process of degradation and dispersion acts to dilute them and dissipate their effects into the environment around them. The problem we're studying appears to happen around the time of the lowest concentrations of neonicotinoids that we expect to see in the environment. So, scientists get quite excited about things like cause and effect, right? So one is meant to come before the other. This doesn't seem to be the case here. This also applies to one of the other notorious problems associated with neonicotinoid seed dressings in other countries, and Don will talk to you about this and why it isn't a problem in New Zealand. Again, when the seed's being drilled, you often get this drift of dust, which can spread onto nearby flowers where bees might be foraging, and it's a way of massive concentrations of neonicotinoids being returned to the hive and causing mass bee deaths. Again, this doesn't happen in New Zealand. If it does, where are the reports? We don't hear of mass bee deaths at the time of seed drilling. The problem seems to be anecdotally related to maize harvest, which, as you can see, occurs in mid to late autumn. So this data this slide I put up earlier with the decay of the neonicotinoids in the environment, this is some American data. We've set out to work out whether this is actually the case in New Zealand. You don't want to take other people's work for gospel. You want to go out there and repeat it yourself and determine whether that's the case experimentally. So with the help of some friendly farmers and landowners, Don jumped a few fences as well, and we set out to collect some soil samples from some maize paddocks immediately post-harvest. Took them back to the lab, extracted the neonicotinoids with some funky chemicals, and put them into this fantastic machine in my laboratory, which is called an LCMS. I'm not going to tell you what that stands for, but there's big words like chromatography and mass spectrometry involved. We quantified 5 neonicotinoid pesticides, and we found only 2 of those. But those 2 that we found, clothianidin and imidacloprid, we found in almost every sample that we analysed at quite low levels. As you look at the bottom, the line in black shows the mean across all the sites that the average concentration we found across all the sites. And again, these numbers are in parts per billion. You can see at the bottom it's about 8 nanograms per gram of wet soil clothianidin and about 5 of imidacloprid. Those are, again, they're pretty low concentrations and slightly higher than has been reported elsewhere. But that doesn't seem to be a smoking gun. That's not the crazy high levels that might suggest that we have a real problem here. So that's what's in the soil. I mean, honeybees don't forage on soil. Is there a way that those residues can get into the honeybee hives? Is there some way that they're being concentrated or collected en masse in a sudden concentrated period of time in a way that can transfer dangerous quantities back to the hives? Well, in all the time that we spent wandering around maize paddocks, we noticed a couple of things and came up with a few ideas that we ought to investigate. If you look at I'll just go back a slide. You see the maize is quite brown, it's dry, it's ready for harvest. It's not actually dead. If you look at the stems are still quite green, they're still wet. I've bent this one over here double, and you can see that there's droplets of sap being released from it. If you break it open, it's actually sweet. You can taste the sugar in the sap. So once the harvest has been through the field, quite often you see something like this, which is a topped-off maize stem, and there'll be a thin layer, there'll be a sticky layer of liquid on the top of this. Now, bees are pretty smart. They're pretty efficient foragers. They're not one to turn down a free lunch. We thought that this could be a way for the bees to accumulate large amounts of neonics and bring them back to the hive, and it's tied in nicely with the maize harvest. With our friends at Waihua Bees put a pallet of hives in the paddock for us, and Don and I put on our high-vis vests and went around dodging harvesters and tractors, surveying the paddocks immediately after harvest, looking for bees foraging on this material, expecting to see something, maybe hundreds of bees, maybe thousands of bees. We saw none. We saw no bees. This does not seem to be a problem in the one site that we studied, but there were definitely bees foraging on that day, and we saw no evidence that this was a way for neonicotinoid residues to get back to the hive. Okay, that's good. A negative result is still a result. We had a good look around the paddocks, and we've looked for some other ways for these residues to get back to the hive. One thing that's been reported recently is that the residues, partly as a result of their systemic nature, they're water soluble, as I said before, they're mobile. They don't stay within the nicely defined bounds of the paddock where they're sown. They actually leach through the soil into waterways and certainly into the margins of the field where you find a lot of flowering weeds growing at this time of year. Here you can see some dandelions, some red clover. We haven't actually done any systematic collection of these plants to look to see whether there are residues in them. This has been reported elsewhere, and it matches the kind of concentrations that you'd see in the pollen and the nectar of other plants, so in the low parts per billion range. Well, what we did do is, fortunately, we got sent a whole bunch of honey samples and bee samples by Paul Badger out in Gisborne, from some hives that he's lost quite recently, some failing hives, and I've again extracted them and analysed them with our fantastic instruments. You can see on the axis on the left that these are, again, nanograms of imidacloprid per gram of honey. And again, this is parts per billion, and they're very low. The average is about 0.1 to 0.2 parts per billion in the honey of hives which were failing, which were dying. So you can see on the other axis where there's 5 sites, 3 sites at Gisborne and a couple of our experimental sites, including the guys at Waihua. So those concentrations in 0.1, 0.2 parts per billion, are those hazardous in nectar, in honey? We think not. I spent a long time carefully poisoning honeybees and bumblebees. We can do the calculations as we did with the pollen. If you're over the maths, you can look at the nice picture of the bee, the nice video of the bumblebee on the right. Sorry, I couldn't find my honeybee footage. I've archived it a little too carefully, and so I've had to put a bumblebee video up there, but you can see there's a syrup tube in the top of its cage. So we were feeding these bees spiked syrup to work out what concentration would kill them, basically. So if we go back to our lethal dose, 4.5 nanograms for imidacloprid, bees consume about 40 microliters of honey a day, so the lethal concentration should be about 100 micrograms per litre. That's 100 parts per billion. In reality, it's not. Neonicotinoids have an anti-feedant effect. So as soon as the bees start consuming some nectar that's got a very high concentration in, it dopes them up. They lose their appetite. They stop eating it. So the lethal concentration is actually much higher, nearly 20 times higher at about 1,700 micrograms per litre, 1,700 parts per billion in nectar. So if we ask what plausible mechanisms there are for neonicotinoids to contribute to hive losses, as far as acute toxic effects go, we have found no evidence to show that they have an effect on the honeybee losses that would tally up with the seasonal timing of the losses that we're seeing. That doesn't mean that they're not a problem. We can't be sure. Toxicology is a very complicated thing. Besides the acute toxic effects, there's a possibility for chronic toxicity long-term exposure to low concentrations to play a role. These can have sublethal effects. It's been acknowledged that neonicotinoids can affect bees' learning, their navigation, their communication, the productivity of the hive at very low doses. There's some good evidence out there to show this. And finally, there's a potential for synergies between neonicotinoid pesticides, other pesticides, and also pathogens. So a synergy is where you have 2 components which work together to create more than the sum of the 2. They have a multiplicative effect instead of an additive effect. So in short, there's no easy answers here. Our work is really targeting the low-hanging fruit. We've gone looking for a smoking gun and not found one. But there's a lot more work to do before anyone can make any conclusive statements regarding the safety or otherwise of neonicotinoids for honeybees. Don, do you want to wrap up?
Well, we need more money to get research. If we're going to get the answer to this question that Neil Mossop originally put to me, we've got to get some more funding. The link between the maize crop and the harvest and the death of hives tends to be a coincidence of timing, probably. We failed to secure any funding in a late effort for this year's research to do replicated trials. We missed out in the NBA Auckland dish out, and we are funding this research completely by the generosity of AUT. So I would like to see Happy New Zealand get a transparent research funding proposal or process underway to support it. I really like seeing Zespri's proposal there that they talked about before. I'd like to thank a large bunch of people that, if we had not had help from them, we would not be here today, and we wouldn't be anywhere near where we are at the moment. Waihua Bees, Barry Foster, Paul Badger, providing samples, the farmers and contractors who are involved on those maize crops. And there's a relationship that I think every beekeeper should try to strive for. Alan Ernest phones up the beekeeper and tells him he's going to start maize harvesting in a couple of weeks, move his hives. That's quite good advice and a good relationship going in practice. And I'd like to thank Chris and Iana for doing all the work that was being done in their lab. I'd like to finish with a poem written by the famous graffiti artist Banksy in 2005. I'm not going to read it right through, I'll leave it up for you to read, but it's a story about a bear and a bee. And the bear watches the bee through the summer, harvesting the honey, and when it gets winter, the bear gets cold, so he thinks he'll go and rob the hive. He arrives at the hive, and what's he find? A dead bee. His excuse was it was a stress-induced coronary incident. Okay, we've found the dead bees. We haven't got the answer exactly why they're dead, but we have to work through. Any questions?
Yeah, Don, I have a question. Last speakers had talking about 1.2 million in research. How much sort of money are you looking at to do this research that I believe, you're only talking 200 hive deaths, but I believe you've got hive deaths that are happening out there in sublethal doses and affecting a lot of hives. Beekeepers are not putting hives into certain maize areas and certain areas. They know they're getting poisoned. Of course, they're not telling their fellow beekeeper because he goes and does the same thing. I think they actually suspect it's affecting many, many hundreds of bees. So what sort of money are we talking for this research?
At the start of the year, we were looking in January for 20,000, which was going to be doubled by AUT, so it would be 40,000 for a four-site replicated search, replicated plotting area, and we had lined up 4 paddocks that we could probably do the work in. So we weren't talking a lot of money, but it was significant at the time, but we had to start early. So we missed the Sustainable Farming Fund shuts off on the 1st of August, so you've got to get your submissions in at the end of July. So when you make decisions, and we only started in September. We started actually in June, but we did in September was when we actually did the jump the fence samples.
Dollar for dollar, so you're looking around $20,000.
Yeah, that's right. But I will say this, what we've learnt so far, that we'll be probably expanding into that program more because we'd want to look at the pathogens effects.
Hi there, Stephen Browning, Green MP. I missed the very start, unfortunately, of your presentation. Probably a bit tired about talking about this issue last night at a public meeting in town. But have you looked at the task force on systemic pesticides findings?
Yes, we've looked at what research has been done on systemic pesticides. What we can't find is, when we look at the crop, we can't find the bees. There's no flowers in a maize crop at harvest. There's no attractive there other than the sugars that may be in the stem. So we know that the sugars are in the stem because we can break the stem, we can see the capillaries where they come up, and we can taste it. You can actually break open the stem and you can taste the sweetness, but there's no bees there.
On that particular one paddock that you've
We've walked that paddock, and John McLeod has in fact walked paddocks in the Gisborne area on the same sort of idea, to try and find the bees foraging. We have heard beekeeper reports that they've seen bees foraging on maize stalk, but we have never had it documented and never seen the photos or the evidence.
And that's, it seems like you're limiting for that acute outcome for that particular time of the year, and the report from Task Force on Systemic Pesticides suggests it's a far more chronic situation that's happening with neonicotinoids.
That could be correct.
Sorry?
Sorry, you?
That seems to be the case. There's evidence to show that there are chronic toxic effects.
And so are we maybe
concentrations
seeing a culmination of some of that? I mean, it's research for you guys to do.
Yeah.
In winter, back in the brood, back in the hive, as opposed to what's happening in the field, maybe.
We don't know.
But we do know from the report of over 1,100 published papers that there's an issue in terms of bee deaths.
Yes, and
So we don't need to reinvent the wheel totally in New Zealand, is what I'm suggesting.
correct, Stefan, but one of the things is that Neil Mossop and the beekeepers will tell you, their bees are not sitting next to the maize crop for the whole season.
No.
Neil's bees were dying when he brought them into this area near the maize crops at maize harvest at that time after they'd come out of Manuka. So they haven't been on that maize crop for the whole length of the season. And that's pretty much normal practice from what I can gather talking to other beekeepers. The bees that you saw on there, they'd been used for pollination and kiwi fruit, gone out to do Manuka, and were back wintering over when we put them on the site.
And what I'm concerned about, what I'm seeing in New Zealand, is the widespread use of neonics over a range of crops, including kiwi fruit, including grapes, and so the swards and the longevity of them means that the neonics are basically totally persistent through the year. So that's the area that I'd be hoping that you'd be doing something fairly quickly. But in the meantime, we are also having maize growers who aren't using neonics and getting very, very good crops. So shouldn't we be pushing the growers to be backing off on neonics in the meantime?
We haven't looked at that, so
We're out of time too.
we haven't really looked at that. We know that the neonics are persistent at low levels, low parts per billion concentrations. Whether that is a problem, that's in the soil again, whether there's a vector for those low concentrations to get into the hives, to have an effect at the times of year where we're seeing the problem, that's work that needs to be done.